Hello, how are you? I hope you're well and looking forward to today's session. The last two sessions have been rather long and quite dense in information, terminology, and I also was under the impression that you were starting to get a teeny weeny bit bored. So today we're not going to go straight away and start with a speech. We're going to do a couple of exercises, if you don't mind. If you find them boring, you can skip them. The first exercise is one I do a lot with my students in the classroom. I've observed that we increasingly have problems to focus and concentrate. So I usually ask them to choose a long word and to spell it backwards. You can use, for example, the word recommendation. So just sit back, don't use your hands, your fingers, or anything at all, and just try to spell the word recommendation backwards. How did it go? Was it difficult? My students usually tell me it is, and they also tell me that they have to visualize the word in their minds so that they can see each one of the letters and spell them in the correct order. If they don't visualize it, they don't imagine what is going on, then they can't spell the word. The same happens with interpreting. This is a very, very good exercise. So, are you ready to go? Yes. Let's go then. Microtubules, DNA, prokaryotic cell, eukaryotic cell, linear DNA, rough endoplasmic reticulum, Golgi apparatus, mitochondrion, bacteria, surface projections, nonpolar molecules, yeasts, fungus, biochemical environments, RNA, microvilli, cell junction, mitotic reproduction, negative charge, diffusion, expanded cell volume, biosynthesis, smooth endoplasmic reticulum, circular DNA, epithelia, basal surface, hemidesmosomes, self-repair, microorganism, endocytosis, single-celled organism, binary fission, protozoa, lysosome, archaea, cytosol, ribosome, organelles, mitotic spindles, protist, to pinch off, vesicle, dynein, peroxisome, flagella, surface pit, cell diversity. So how did it go? Did you feel nervous? Did you get all worked up? Never mind. It was just a warm-up exercise, and if you managed to do 1 out of 2, you did really, really well. You're fabulous. And now we're going to proceed to today's speech. And I'm going to start now. Good evening, ladies and gentlemen. This is the third in a series of 7 lectures on cell biology and genetics. If I'm not mistaken, the 2 prior speakers covered the introduction to this topic, and as a result, I believe you've already heard about prokaryotic and eukaryotic cells. I have been informed that they were very long and difficult speeches, so today I will just talk about cell diversity very briefly. Researchers believe that multicellular organs evolved because similar types of cells combine together to form a single organism. They also combine and form the four main types of tissue: epithelial, muscle, connective, and nervous. Epithelial tissues refer to groups of cells that cover the exterior surfaces of the body, line cavities and passageways, and form certain glands. Connective tissue binds the cells and organs of the body together. Muscle tissue contracts forcefully when excited, producing movement. Nervous tissue is also excitable, but generates and propagates electrochemical signals in the form of nerve impulses that communicate between different regions of the body. And now we're going to talk about cell differentiation. What is cell differentiation? It is the process that a young and immature cell undergoes until it evolves into a mature cell thanks to the expression of successive cascades of proteins regulating the DNA in each cell and differentiating them from each other. An interesting fact is that over 200 types of cells are identifiable in human tissue, and they have different structures, functions, and chemical metabolism. But we have to remember that there are other elements that play an important role in cell differentiation. We're talking about gene structure and environmental factors. Gene structure because it determines the cell type and the physical attributes of the animal. Environmental factors because if they undergo changes, they can affect the release and production of hormones. All cell types derive from a single cell, the zygote. The zygote is totipotent and will eventually become an entire organism. Genetic material is not lost during human development. Different cells arise as a result of human development. Even the largest animal on Earth, the blue whale, starts with a single cell. That is the beauty of nature. It never ceases to surprise us. I would like to conclude my speech by talking about the concept of cell memory. Even when a cell is removed from its normal environment, the differentiated cell and its progeny or daughter cells will retain many of its functional characteristics. This is because in 2013, a possible mechanism was found for how this cellular memory works and how it helps the cell remember the order that existed before the cell divided. As I think we're all very tired, it might be a good idea for us to visit the poster exhibition outside and discuss any questions you may have informally. Ladies and gentlemen, thank you very much for your attention. So how did that go? The speech was much shorter today, wasn't it? I think it was good. And if you did the exercises, I hope you had a good time, because I'm not here to bore you. I'm here to help you and to try to study interpreting having a lot of fun. It will be better as time moves on. Thank you very much.